Okay, so our next speaker will be Amy Griffin, who will speak about the prize in physiology or medicine. Amy's been a faculty member at UD for 7 years, and she has just, within, I guess, the last 2 months, been promoted to the rank of associate professor in the Department of Psychological and Brain Sciences. She earned her PhD in psychology in 2003 from Miami University in Ohio, and then did a postdoc in the Center for Memory and Brain at Boston University. She came to UD immediately after that. So, Amy?
Well, good evening. It's very much an honor to be here. Here's the clicker. I'm going to start this talk by asking you to do something very simple. I want you to think about how you got here this evening. Okay? So, if you're familiar with campus or you've passed by this building a few times, you may not have had any trouble. If you're not familiar with campus, you may have used your smartphone or even a good old-fashioned map to get here. So if you are familiar with campus, you've been to this building a few times, you've passed by it on your way to somewhere else, you may not realize it, but you use an even more sophisticated GPS system. The GPS system that's been built into your brain and nurtured by evolution. So even though understanding how the brain helps us find our way in space may be interesting in its own right, even more fascinating is the process of navigation is linked to the way that we store memories. So tonight, I'm going to be telling you about the Nobel Prize in Physiology or Medicine that was awarded to 3 neuroscientists, the husband and wife team of Maybrit and Edvard Moser, and their former mentor, John O'Keefe. And they won the Nobel Prize for discovering the positioning system of the brain. So I'll start by giving you an overview of the techniques that were used in these kinds of experiments. And I'll explain the phenomenon and then take a few minutes to talk about how this work influenced my career. And finally tell you about the award winners themselves. So brain cells, or neurons, represent all kinds of information by emitting these really brief 1 millisecond spikes of activity called action potentials. And this is how neurons communicate in a number of brain regions. In particular, the regions responsible for spatial navigation are mainly these 2 interconnected structures located in the medial temporal lobe of the human brain, called the entorhinal cortex and the hippocampus, my favorite brain structure. So to record these action potentials, rats are implanted with an array of microelectrodes. You see an electrode aimed at the hippocampus, the hippocampus is right here. And these microelectrodes are thinner than a human hair. So an electronic device connects the microelectrodes up to a computer, and that allows the experimenter to listen in on and record the brain activity. These recording devices are lightweight and easily carried around by the rat. Here's a rat. So it's possible to link together in time the position of the rat and the spiking activity of hundreds of neurons. And if you hook up the electronic device to an amplifier, it kind of sounds like popcorn popping. So in this short video, which I hope we'll be able to play, you'll see a rat running around on a T-maze in a figure-8 pattern. Yeah, and what you're hearing is action potentials from a cell in hippocampus. And what I want you to do is tell me, what part of the maze do you think this cell is responding to? So what do you think? We can pause it. There we go. We'll go back. Anyone? A left turn, you think? Yeah. Anyone else? It's the place in the environment, so it's responding when he's running up this central stem, right? And you're right, it's only on left turns. So it's really interesting. So this is a variant of the phenomenon that was discovered by John O'Keefe, the hippocampal place cell, and I'll describe that in detail in a moment. Well, I'll go back here. Psychologists have been studying spatial navigation for many years. So initially, the predominant view was that behavior could only be studied through stimulus-triggered responses. But however, in 1948, Edward Tolman proposed the idea of a cognitive map. That is, the brain contains a kind of map of the environment, and then experiences can be encoded on top of that map. And support for this idea came years later in 1971 with a remarkable discovery of cells within the hippocampus that coded spatial location, just like the one we just saw. So John O'Keefe, along with his student John Dostrovsky, published a paper in a rather obscure journal, reporting that they discovered a type of cell located within the rat hippocampus that was selectively active whenever the rat visited a certain part of its environment. And they called these cells place cells, and the location of the rat where the cell was most active, the cell's place field. And looking at the recording enclosure from above, this is a recording from a hippocampal place cell. The places visited by the rat are plotted in black, and a red dot is placed at the location of the rat's head every time the neuron emits an action potential. So you can see the place field would be right here. So by recording from multiple cells, they discovered that different cells, shown here in different colors, code different spatial locations. So theoretically, if we recorded from all of the cells in hippocampus, every place in the environment would be represented just like a map. If you move the rat to a different environment, a different set of place cells would be active, suggesting that the hippocampus constructs a different map for each situation that the rat encounters. So a few years later, in 1978, O'Keefe and his colleague Lynn Nadell published a highly influential book called The Hippocampus as a Cognitive Map. And this book was on my walls. Their claim was that the place cells provide the animal with a dynamic, continuously updated representation of space along with the animal's position in space. In other words, Tolman's idea of the cognitive map was correct. But one key question remained. How is a spatial code generated by the hippocampus? So a major breakthrough in answering this question came decades later, in 2005. The Mosier group, Edvard and Maybrit, discovered a spectacular cell upstream of the hippocampus in the entorhinal cortex that they later called the grid cell. To illustrate the properties of these grid cells, again, the places visited by the rat are plotted in black lines, and the red dot is placed at the location of the rat's head each time the neuron emits an action potential. And this is a neuron recorded from the entorhinal cortex. After a few minutes, what they noticed is these dots began to show a pattern. The clusters of dots form the vertices of a grid of equilateral triangles. So we can draw a triangle between all the vertices. Now, by contrast, if we look at that place cell we saw earlier, there's usually only one cluster for the entire environment, and even when there are multiple clusters, there's really no regular pattern to their arrangement. And this remarkable hexagonal pattern has been likened to longitude and latitude coordinates on a map. So the grid cells provide us with a coordinate system that helps us to judge distance and navigate. In just 9 years, there have been almost 200 papers published on grid cells, which gives you a sense of the impact that this discovery has had on our field. So since the discovery of grid cells, the Mosier group has focused on learning how grid cells communicate with other types of neurons, including place cells and two other types of cells, border cells, which they discovered in their lab as well. And these signal the boundaries of the environment. And then head direction cells, which are most active when the animal faces a certain direction regardless of its spatial position. essentially acting as an internal compass. They're now interested in uncovering how these neuron types work together to accomplish spatial cognition, memory, and decision-making. So at this point, maybe you're asking yourself, how did these findings relate to humans? And while it's true that the vast majority of these studies have been done on rats, place cells and grid cells have been found in every species that's been studied so far, including mice, bats, monkeys, and even humans. A group at University of Pennsylvania, led by Michael Kahana, has discovered a way to record single neurons from the hippocampus of the entorhinal cortex of the human brain. They use a subject population of epileptic patients who have electrodes implanted deep in their brains in order to localize the seizures. In Kahana's experiments, these patients navigate around a virtual reality environment as their brain activity is recorded. And this group has found that place cells and grid cells in the human brain have identical properties to those found in the rat. Now, long before the discovery of place cells, we knew that the hippocampus was critical in forming memories. And we owe this insight to one particular patient known by his initials HM. In 1953, HM had surgery that removed most of his hippocampus in order to cure his severe epilepsy. And even though the surgery alleviated his seizures, the removal of the hippocampus left him with this unfortunate side effect of being unable to make new memories. And this type of amnesia has been depicted in many popular films, including 50 First Dates, Memento, and the favorite in my household, Finding Nemo. So animals and humans with hippocampal damage lose two critical functions: the ability to find their way and the ability to form new memories. So insight into how the brain constructs complex mental representations of the world, like memory and spatial location, can potentially lead to interventions that help to millions of people that are suffering from brain disorders. The Obama administration has even recognized the importance of funding basic neuroscience research by establishing the National Institutes of Health Brain Research through Advancing Innovative Neurotechnologies, the BRAIN Initiative. The entorhinal cortex and the hippocampus are the brain regions that are first damaged in Alzheimer's disease. In fact, one of the early symptoms of dementia is getting lost in a familiar environment. So the better we can understand these circuits, the closer we'll be to understanding what exactly goes wrong in memory disorders and possibly develop therapeutic interventions that can slow or reverse this process. And the popular press has been describing these discoveries as finding the GPS system of the brain. And for those of you who use your cognitive map to get here tonight, as you walk toward the ICE building, your grid cells helped you to judge distance and navigate. The head direction cells acted as an internal compass, telling you which direction you were facing. And the place cells helped you use landmarks and previous experience with navigating around this campus to find your way. So I thought I'd take a few minutes to tell you about how these findings have impacted my career. So I'm interested in the brain mechanisms of memory function, particularly episodic memory. And this is the memory that allows you to remember events in your life. And typically when we remember an event, you perform a sort of mental time travel, recalling all the details, and you recall the when and the where. And the brain circuitry that gives us a sense of space has a substantial overlap with the circuitry that's critical for episodic memory function. And perhaps this is because both functions require that multiple modalities, so sight, sound, touch, smell, be integrated together. So I've been recording hippocampal neurons for over 17 years, at first as a graduate student and then as a postdoctoral fellow, and now in my own lab at UD. Like so many people in my field, my research, questions, and ideas would not have been possible without O'Keeffe's discovery. For example, one major finding from my lab is that hippocampal place cells have different firing properties depending on what task the rat is performing. Critically, the tasks are performed in the same room, in the same environment, suggesting that it's the behavioral context and not simply the spatial location that's an important factor driving place cell activity. So the last part of the talk, I'll just tell you a little bit about the people behind the prize. John O'Keefe received his PhD in 1967 from McGill University. He then moved to University College London, where he's been ever since. He's trained many successful neuroscientists who have continued his work by making substantial contributions of their own. A former postdoc, Colin Lever, called O'Keefe's discovery a cognitive revolution. And that's because in 1971, the scientific community wasn't ready to consider that animals could have a mental representation of the world. So it took several years for O'Keefe's findings and ideas to be fully appreciated. So two of O'Keefe's postdoctoral trainees are none other than May-Britt and Edvard Moser. May-Britt and Edvard earned their PhDs in 1995 from the University of Oslo. After doing their postdocs at the University of Edinburgh and at UCL with John O'Keefe, the Mosers returned to Norway with faculty appointments in biological psychology at the Norwegian University of Science and Technology, or NTNU, in Trondheim. Additionally, the Mosers are co-founders and directors of the NTNU Center for the Biology of Memory and the Kavli Center Institute for Systems Neuroscience. So former trainees of the Moser Lab describe them as generous mentors and just down-to-earth regular people who like to go fishing and hiking on the weekends in the beautiful countryside of Norway. May-Britt and Edvard call the rats in their experiments their most important collaborators, which I love. And they also show a great deal of gratitude for their entire research team. So in summary, John O'Keefe discovered the place cells in the hippocampus that make up the cognitive map. And over 3 decades later, May-Britt and Edvard discovered the coordinate system of the brain, the grid cells. Together with head direction and border cells, this neural circuit serves as our internal positioning system. So as you leave this building tonight, I hope you will give some thought to the brain mechanisms that help you find your way home. Thank you.
There's a question in front here. I'm going to ask everybody who has a question to speak into a microphone so that it gets picked up on the videotape.
So there's a lot of phenotypic variation among humans in their ability to navigate. Has anybody looked at the genetic variation, either in rats or humans or any organism, in this ability to navigate? Well, they're mental maps. And
Yeah. I'm glad you asked that question. There's a really interesting study that was done. It's not genetics so much as experience. There was an experiment done on London cab drivers. And it turns out that their hippocampus is much larger than the rest of our hippocampi. So the experience navigating around London, because these London cab drivers need to be highly experienced, highly trained, and gather a lot of experience about this environment, their hippocampus is much larger. So does that answer your question a little bit?
I think there's a lot of phenotypic variation that sort of runs in families, for example.
Right.
That suggests that there's a genetic basis for differences among individuals, or at least in part.
Mm-hmm.
Sorry. Any other questions?
Other questions?
Yeah. Why don't you come up and get Now it's on.
This is on. So the animal models that you mentioned earlier all really live in a two-dimensional world. Has there been any research done on, say, maybe aquatic animals, maybe fish, who really live in a more three-dimensional map?
They're actually, it's not fish, but they've done an experiment. Not a whole lot came out of this, but it was a really, really interesting, cool thing to do. They put rats in space, and they recorded place cells in a non-gravity environment. And I mean, the only thing that really come out of that experiment was, yes, you can get place cells in 3D. But no, they haven't recorded in fish or aquatic animals.
But they did bats, right? So,
Bats, yeah, so that is 3D space. That's a good point.
are
So,
there
yeah,
different kinds of cells, or is it pretty much the same picture, just translated to 3 dimensions?
It's translated to 3 dimensions. The first discovery of place cells in bats were in crawling bats. So they're crawling around, and people said, well, that's not really ecologically valid to what the animal does. And so they came up with a way of having these kind of wireless systems, recording devices, hooked up to the bats, and now they are recording from bats. Malcolm Ulanowski at the Weizmann Institute does that, so it's really fascinating work.
Any other questions? The initial discovery that you mentioned was back in the 40s? Was that?
71.
I thought you said there was this obscure paper that was published.
No, so
Back in the Dark Ages.
in 48, that was the first, so that was when Tolman proposed this kind of revolutionary idea of a cognitive map. And then it was 1971 when the first place cell paper came out.
Okay. But there was this case of this patient who had lost his hippocampus.
That was in the 50s. That was in 1953.
Did he have difficulties, navigating as a result of that? Did they start to make
Yes.
connections? I mean, I'm kind of wondering why it took so long to make that connection.
Yeah, I think it was because people weren't ready to see it. The community wasn't ready to see it. But HM did have trouble navigating. He could navigate around his environment, his home, after lots and lots of practice. But to learn a new place, he was not able to do that.
Great. All right, well, let's thank Amy again.